So my name is Fateeha Furqan. I'm one of the third-year hematology oncology fellows at the Medical College of Wisconsin, where I've been focusing my research on lymphoma and CAR T-cell therapy, and I've been working with Dr. Neerav Shah, who's been my mentor during my fellowship. This project that we did, this is a retrospective study in which we included 11 patients who got Pirtobrutinib, which is a newer non-covalent BTK inhibitor prior to CAR T-cell therapy. So these patients received Pirtobrutinib at least 4 weeks prior to apheresis because we wanted to study how the BTK inhibition affects the manufacturing as well as the outcome of the 2019 CAR T-cell therapy. This was a part of a clinical trial. The 2019 CAR T-cell therapy was a part of the clinical trial, so the patients got 2019 CAR T-cell therapy as part of the clinical trial. The reason we did this study is because there's some recent data showing that combining BTK inhibitors with CAR T-cell therapy improve outcomes. Most of this data comes from CLL, and in CLL they've combined ibrutinib, which is a covalent, not very selective BTK inhibitor with the anti-CD19 CAR T-cell therapy. The preclinical data has shown that both ibrutinib and zanubrutinib improve the CAR T-cell therapy, so they decrease the expression of exhaustion markers and improve CAR T-cell therapy effect overall. And then there's clinical data in CLL which has shown that patients who receive ibrutinib with anti-CD19 CAR T-cell therapy did pretty well, although they are small studies. But in these studies, there was an increased signal towards toxicity, particularly cardiotoxicity, which can be really high when you combine this with a toxic therapy like CAR T-cell therapy. So that's why we chose Pirtobrutinib, which is a highly selective BTK inhibitor, and it doesn't have a lot of cardiotoxicity like the rest of the BTK inhibitors have. So the patient received Pirtobrutinib at least 4 weeks prior to apheresis, and then went on to get apheresis, and then went on to receive the 2019 CAR T-cell therapy that we have at Medical College of Wisconsin as a part of the clinical trial. There were 11 such patients. Overall histologies, most of them were either mantle cell lymphoma or Richter's transformation. All patients except one met the target dose of the CAR T-cell therapy. We did preclinical studies, so we studied the immunophenotype of the CAR T-cell therapy, and we studied the polyfunctionality and the polyfunctional strength index using isoplexis. And we compared these patients to the patients who just got the CAR T-cell therapy, the same 2019 CAR T-cell therapy without the Pirtobrutinib. So what we found was, in terms of immunophenotype, there was not a lot of difference between patients who got the BTK inhibitor prior compared to those who did not. So what we looked at was naive naive T cells, and then the ratio of terminally differentiated T cells, and they were similar in both these patient groups. We compared the apheresis product as well as the final CAR T product, which was given to the patients. Then we did polyfunctionality and polyfunctional strength index using the isoplexis and the IsoSpeak software. This generally measures the cytokines which are released by CAR T-cell therapy when they are stimulated by CD19 positive cells. And what we found was that patients who received Pirtobrutinib prior to the 2019 CAR, they had a higher polyfunctionality and a higher polyfunctional strength index, although this was not statistically significant, but it did point towards that there's a signal that these patients are more functional, or these CAR T cells are more functional. Then in terms of the clinical outcomes, the overall response rate was around 80%. There were not a lot of side effects. So around 3 patients developed CRS and ICANS, and all of them were grade 1 to 2. 3 patients developed IECHS, or the HLH-like phenomena, which happens with CAR T-cell therapy, which is probably related to the underlying histology. So what we found was patients who have either Richter's transformation or CLL, in our previous clinical trial, tend to have a more HLH than patients who have other histologies. So we think it's related to the underlying histology rather than the BTK inhibition itself, although they're very small numbers. We have a median follow-up about 13 months, and in the follow-up, there are 3 patients who died, one because of disease progression, one because of COVID, and one because of Guillain-Barré syndrome. And then, so to conclude, we think that BTK inhibition just BTK inhibition with Pirtobrutinib can improve the CAR T product itself in terms of the polyfunctionality and can eventually improve outcomes of CAR T cell therapy. That's why we are opening up a clinical trial at the Medical College of Wisconsin, in which we are going to combine Pirtobrutinib with the 2019 CAR T cell therapy that we have as a bridging therapy, and then we're going to also continue the Pirtobrutinib as maintenance, and hopefully this trial is going to open in July.